Science of Feeding Tomorrow. Pathway Intermediates.
It's very nice to meet you. My name is Hanbei Ni from Pathway Intermediates. We would like to welcome all of you joining us today from all around the world. As you know, although many of us are forced to stay indoors and most of our travels have been suspended, we have wisely adapted to this post-corona way of life. And thanks to the development in IT technology, we can overcome many physical obstacles and meet up using video conference tools like the one we have set up today. In an effort to reach out to our global partners and friends in the industry, Pathway Intermediates has arranged a series of webinars that we will continue throughout the rest of the year. Now, today is the first webinar of the whole series, and we are very fortunate to have Professor Simon Davies as our invited speaker. As many of us know, Simon is a world-renowned professor in aquaculture. To give us a very brief introduction of Professor Simon Davies, he has a background in biochemistry and agricultural biochemistry and fish nutrition. He has been working for a very long period of time as professor at the Plymouth University and is currently the professor of animal biosciences and aquaculture at Harper Adams University. Also, Simon has been our consultant of Pathway Intermediates for our Aquaculture global platform. He's one of the most productive person that I know, with more than 200 peer-reviewed scientific papers, book chapters, and presentations. He has also produced many more students, such as more than 36 PhD students and more than 350 master's students. And these numbers are still counting. Well, as I introduced you, you are a world-renowned professor and a scientist in the field of aquaculture. We are very fortunate to have you with us this morning. And so I believe that everyone joining us today will also agree. And it's very fortunate that you will take us back to school, to give the general overview about the warm water fish nutrition.
Okay, well, thank you very much for those very kind words, and it's nice that you've acknowledged my success, but a lot of it comes from just simply getting older as well. That's how you end up doing so much work when you get actually older. But thank you very much for your kind words, and yes, let's get back to school together. And this technology is a very interesting way of doing it, and more professors are now in academics rely so much on this for the future for our students as well. So thank you very much, Hanbei. So I'm going to share the screen with you from my end, I hope. That's going to work. So let's see if we can do that. And then I can choose my topic and then go to the from the beginning. And now, can you all see that successfully? Is that good? Yes, thank you very much. Well, I was asked to talk about specifically focusing on warm water fish species. And I have to say that this is quite a challenging thing to do, because we talk in general about fish nutrition. When we address aquaculture nutrition, we tend not to differentiate too much from the textbooks. And textbooks and documents cover most of the fish together, and we go by species to species. But to pull out the warm water aspects is an interesting thing to do, and that's what I've tried to do in this address today. Now, of course, there are many different classes of species of fish that can be found in the warm water domain, both freshwater, a lot of freshwater species like tilapia and mirror carp and pangasius. But of course, there are many others, and I'm probably not doing justice to many other of the marine tropical fish. We also call these warm water fish tropical fish, of course, and compared to the temperate species. But there's gray lines. There are gray lines. And so there's overlap because the temperature tolerance of some of these fish can be quite adaptable. So, of course, we have the freshwater domain, the brackish water, and the full-strength seawater species. And each one of those areas has their own challenges. But just to remind you, the tilapia is one of the fish I'm going to focus on because it's a very important species. The global trend in the green bars you see there is that tilapia is reaching about 6-7 million metric tons. The price of tilapia is variable, but it's kind of falling a little bit as we produce more tilapia around the world. But of course, there are lots of issues with tilapia farming, with potential pathogens and diseases and emerging issues, which I'm also working on. And the major area of tilapia production is Southeast Asia, but Egypt is a major producer in the Middle East. To my Middle East friends, tilapia is a very important species in Egypt. And of course, in Thailand and China particularly, tilapia is a large producing nation is China and Vietnam and Indonesia. So I don't want to miss anybody out. Okay. And we have to ask a basic question because Hambai said, let's get back to school. Well, nutrition is the ingestion, digestion, and absorption of food. And fish nutrition research has advanced considerably over the last 30 years to 40 years. And so much has moved on since the days of the great John Halver and other very prominent fish nutritionists. Professor Halver actually sat in this office in 2001. So he was the pioneer of fish nutrition research in the beginnings. And I have his textbook here next to me. Over 50 species of fish have been studied in depth, with much now on carp, tilapia, and of course, catfish, especially Pangasius. We use the term catfish rather generically, but of course, there's the African catfish as well, which is a very important example of warm water fish species found in Africa, in Nigeria, and produced in South Africa. And there are Asian versions of Clarius, another important fish. And of course, the warm water marine, we have cobia, grouper, tuna, and many, many more examples. So I don't want to be unfair to the other species as well. Now, fish nutrition is about, of course, growing the fish. Our challenge is to grow fish. Fish growth is very important. And we have to bear in mind that fish are poikilothermic animals compared to endothermic animals like mammals and birds. And they are subject to a variable temperature, and therefore their metabolism is very strongly linked to temperature. So water temperature is a very important criteria, of course, in warm water fish, ranging around 25 to 30 degrees. We wouldn't want to go outside those extremes. Body size, the stage of development, fry to fingerling, juvenile, on-growing. Body size, of course, influences the accumulation of body mass and tissue. And age, the chronological aspect of the fish is related to body size, and physiological changes as well. And there are many other environmental factors that impinge on this, but we mustn't forget oxygen. Oxygen is a vital component in the assimilation of the nutrients. And perhaps sometimes we tend to forget the important interplay between good nutrition and oxygen consumption, something I'm looking at now very closely in my research. And warm water fish are no different to the temperate cousins, but we have an optimum standard temperature which is needed to sustain good growth, and we call this the standard environmental temperature, SET, which I've highlighted there. In yellow, I've tried to highlight some of the more important things. Optimum temperature is found when maintenance requirement for energy are relatively lower and appetite is higher. During that bandwidth, we get the best growth rates of the fish, which you'll see in a diagram scheme I have to show you. The National Research Council recommendations for salmon and trout is around 12 to 15 degrees. But when we get to the tropical fish or warm water species, then channel catfish and tilapia can attain a much higher temperature, and the optimum for them is around 28 degrees Celsius for tilapia. Channel catfish, of course, are grown very much in the southern parts of the United States and are very popular there, and you mustn't confuse them with the other generic catfish terms. Temperatures, therefore, where the metabolic rate for each species is optimal for energy utilization and protein synthesis. there's a 2.5 increase in growth, roughly, for every 10 degrees Celsius rise in temperature. And that is an important thing to bear in mind, that we are increasing the scope for growth rates at every 10 degrees Celsius rise in temperature, up to a maximum. The amount on environmental factors, such as on fish growth, would involve water flow rates, chemistry, all these things can affect the metabolic rate. Oxygen availability and the metabolic demand challenge of warm water fish is very different in many ways to cold water species, because as you elevate the temperature, the amount of oxygen that the water can carry is reduced. Therefore, there's a kind of trade-off between oxygen demand, metabolic rate, and how much we can then deliver to the fish for metabolism. Of course, light is very important too. The ratio of darkness to light, diurnal changes in light and rhythm can also affect this. And warm water fish may do better with extended light regimes because they're more used to that in tropical climates. So there's an optimum requirement in production of cod and salmon and pre-smolt production. We know this to be true. I think that's another area to be investigated because feed intake and lighting is such an important aspect of growth, especially in recirculation systems and RAS technology, which is becoming more of an important area now that we can't discuss today, but bear it in mind. Overcrowding, undercrowding, all these factors create stress, but we are lucky that some of those warm water species like tilapia do very well at high stocking densities and trigger off a better feed intake. Right then, now this is a very interesting scheme. I hope you can see that, but it breaks down temperature and growth rate in relation to breaking down the energy balance and demand for fish. The Cmax is the maximum feeding rate, and you can see there the top profile. It goes up with temperature. Temperature plays a role in the breakdown of the energy partition, and SDA, which is the digestion or specific dynamic action, the amount of heat produced during the process of digestion is influenced by temperature. The amount of fecal production, urinary production, ammonia production, which all leads to energy losses, will be affected by temperature. And this profile was designed really to cover most species, but the warm water fish will be shifted to the right in terms of those profiles being shifted. And we can see there that perhaps the maximum benefits of temperature revolve around, say, 24 degrees Celsius there for a lot of fish. Warm water species have their optimum around 24 degrees Celsius. But during aquaculture production, we may be under that temperature or even higher, but we have to be very careful because at the far right, sorry, of that screen, you might reach lethal levels where we can't supply enough oxygen and the energy demand is too high. So there's this optimum feeding rate and scope for growth. And the scope for growth, the metabolic scope for growth, defines the capacity for the production. And you can see the bottom red line there, if I can see carefully myself, that's the standard metabolism of basal metabolism. And you can see that that will go up with temperature increasing, but the metabolic scope, which is the maximal aerobic metabolic rate at a given temperature, will have an optimum. And during that gap there, you can see that gap is where you would grow your fish optimally. And for carp is around 20 degrees, and for tilapia and other species, it would be higher. I am not mentioning prawns today because this is a totally other ball game, is to look at shrimp. But we'll do that for another day. But they also come into this category. Okay, and the metabolic rates can be affected by spawning, seasonal and physiological changes. We can expect higher rates in the summer when spawning, and lower in the winter when resting. So we can see that production is linked to temperature, and we have to accommodate our nutritional requirements to those aims. The optimal feeding of fish is necessary, and we mustn't feed them over the top, otherwise we'll be wasting the energy, and we have to be careful that we get that balance correct. Okay, I don't know whether you can see me in the corner there as well. I'm not sure during this meeting we can just see the screen, my shared screen, or I think you can see me as well. Now, the energy flow through the fish is a standard breakdown you see in all animals. but we have to look carefully at the wastage of energy. We feed the fish the gross energy, the feed intake. We have the digestible energy there. We have the losses in feces, which gives us our digestible energy value. And then we can have the metabolizable energy from that. And we have to allow for the wastage of energy from the urinary losses, which fish do produce, and mainly gill losses through ammonia. And the recovered energy we term the net energy. Now, the net energy system exists for a lot of animals, but we are very much behind in fish nutrition in terms of having detailed net energy requirement data. And that's a challenge for the future. So we work with the metabolizable energy, but very often the database relates to digestible energy. And we talk a lot about the digestible energy for fish from the feed, supplied by the nutrients, of course. Now, the digestive model for feeding varies enormously between different species. And because we're operating at a higher temperature for warm water fish, we would have and expect maybe different ratios of nutrients being supplied for lipid metabolism, for intermediary metabolism, which is at the center there. This is a very important area where all the nutrients are conveyed for different purposes to transfer nutrients around the body. Intermediate metabolism is at the fundamental wheels of biochemistry. Protein metabolism feeds into this, lipid metabolism feeds into that, and glucose metabolism, which is going to be quite important for warm water fish, because many of them do very well with carbohydrates compared to other species. Carbohydrates feature quite strongly in providing the energy there, as well as amino acids for carnivorous fish, and fatty acids can be used as well for both energy and for essential purposes. And all that leads where we have a very complex homeostatic control system, a pink box there, but we have the body mass then would be the end result of growth and nutrient retention. So there's all potential for losses there and inefficiencies. So energy requirements for warm water fish are high relatively, and they are required for system maintenance, digestion, and tissue synthesis. Maintenance requirement must be met before growth. That's fundamental. Low maintenance requirement compared to terrestrial vertebrates, so 5 to 6% compared to what birds and mammals might want to use, because we have to generate heat. Heat production in terrestrial animals is a major source of that energy, but we don't have to worry too much about that in fish, because you know the metabolic rate for diversion to temperature control and regulation is obviously not there. So we have less expenditure for supporting fish in terms of locomotion and body temperature maintenance, which makes fish a very efficient animal in many respects for feed conversion. Okay, now then, so energy, of course, is lost to the digestive processes, as I mentioned. We do generate heat production in a fish, but it's dissipated very quickly to the environment. But it's possible to measure the heat loss if you have the precise equipment for that. But we tend to look at oxygen consumption rate and indirect calorimetry to measure the heat losses and energy utilization in fish. So culturists are very concerned with efficiency of conversion to biomass. So all of this relates to high, good efficiency, and therefore in our measurements, low FCRs. We're always aiming for very good feed conversion and maintaining low FCR. Okay, and here we see an important criterion, and that's the quantitative energy requirements for fish and protein to energy ratio. It's important to get that ratio correct. We express our nutrients relative to the energy intake. So by doing this, we can actually link nutritional requirements, and I've got protein there, but we can have amino acids, we can have other nutrients as well to link that to energy requirements. And you often see data for the protein to energy ratio, or digestible protein, DP to DE ratio, for fish species of any kind. But we need much more data for warm water fish species compared to trout and salmon. And I don't want to get too detailed back to our high school anatomy lessons for fish, but I'm sure some of you out there in the global domain have done fish dissections, much more perhaps than me, but I have done my fair share. And so one has to know our way around the fish. And the trophic system of the fish, it depends on its anatomy, and we must know more about the anatomical detail of fish when we design and understand better their nutritional requirements, especially when it relates to feed presentation, and that's another issue, pellet design and that kind of thing for optimal feed intake. But I just want to remember and refresh the digestive anatomy of fish varies enormously. They're very diverse, ranging from simple stomached fish with short guts like the trout and the carnivorous fish, even the tropical marines. Then we have the omnivorous catfish with this is the channel catfish with a very pouch stomach, a longer gut. And then we have the carp, the catfish, and then the carp, and then they are omnivorous fish with a much longer gut length relative to the body length. And then we get to species that are microphagic and feed on small particulate items. They have a very long gut, and they may have special accessory organs to break down, like the muscular gizzard, analogous to what you find in poultry, to break down particles of food. But we have to remember that that's very important stepping stone in our understanding of fish nutrition. But the fish have an esophagus, they have a stomach, they can be quite distinct and large and developed in carnivorous fish, small in herbivorous fish, like I mentioned. Some species have an extra pyloric cecum to increase surface area, like in salmonids, but we also find those in certain warm water species as well. And the intestine is not just an absorption mechanism for absorbing nutrients, but the intestine, I should say, has many other functions as well, immunological as well as nutrition. So we need to look at the whole system, and the accessory organs that fish have are very analogous to what you find in higher vertebrates, but in warm water species, especially in some other species like carp and tilapia, the liver and the pancreas plays, of course, very important functions, but the pancreas produces enzymes which are very much important for digestion as well as the stomach in some species. So they produce proteases, amylases. Some species may have chitinases, but that's debatable whether that comes from the diet or whether from microorganisms, and lipases to break down lipids. A very important area for pathway intermediates is lipases and lipid metabolism. Okay, so these are very well developed. And let's get to the nutrient requirements. And proteins, fats, carbohydrates, vitamins and minerals are all required in a balanced way. And these are no different to any other animal species, but of course there will be species-specific requirement differences for different fish. And the proteins and their role in fish is really well understood, but they are major nutrients for growth and energy as well, especially for carnivorous fish. They require protein to be diverted into energy utilization, and we are trying to get that balance better so we don't waste protein for energy, but we can use other nutrients. But gluconeogenesis is a process for de novo synthesis of glucose, and carnivorous fish do need glucose, and they're like diabetic mammals in some ways. And there are many examples of that in the warm water species, in the marine carnivores. Amino acids can be found in, of course, natural environment, natural foods, and commercial feeds have to have these amino acids and proteins delivered in the component of the feed. And they are used primarily in tissue repair, growth, and endogenous turnover of nutrients in maintenance. And so when they're in excess, they are utilized for energy, and they can be used to synthesize fat for storage through the TCA cycle, through the intermediary metabolism, which I mentioned earlier. So there are relative expensive components of the feed. We're always trying to address the protein balance to make it more efficient. Larger fish have a less of a need relative to juvenile fish, especially the fry and the fingerlings have a high protein to energy requirement. Protein requirement will increase in water with water temperature, and protein requirements will rise for warm water species relative to other fish. So they do need protein at a higher level for higher metabolism. Mixtures of animal and vegetable proteins can guarantee complementary balance of essential amino acids. There are essential amino acids, and fish meal is a very good source of protein for fish. And our challenge is, of course, to find alternatives to that, which is something we shall talk about later on. But just to remind you, there are 20 amino acids in nature, and 10 of these are what we would call indispensable or essential. Depending if you're in America, they like to use the term indispensable amino acids. And there are some very important ones that are perhaps even more important than others, but there are 10 essential amino acids. And some of the other 10 non-essential amino acids may be conditionally important under varying conditions, like glutamic acid, for instance, is very important for gut health, but it's a non-essential amino acid and can be synthesized. So there are these amino acids of great consideration in feed formulation. And here they are. They make up the protein in the chain. You can see there the primary structure of a protein. The protein chain is the primary structure. Proteins are important in terms of tissue structure and transport proteins. They vary in shape, and they have secondary, tertiary, and quaternary structures, like, for instance, hemoglobin. There are the 10 essential amino acids, and we use abbreviations for them there. There's phenylalanine, histidine, isoleucine, lysine, and methionine, which are rate limiting for many species of fish. They are very limiting in some conditions, especially they are lower in soybean meal, methionine, and lysine is critical and is limiting in corn gluten and wheat gluten proteins. Okay, and these can be produced in crystalline forms to supplement our diets, by the way, to formulate the diets more efficiently. Warm water fish tend not to utilize crystalline amino acids as well as cold water species, because the amino acids in a crystalline form rush into the system and do not match the deficiencies of whole proteins. So that's just a point to bear in mind. And this is a nice way of looking at amino acid requirements by using a beer barrel analogy. The staves in the barrel will vary in height. The amount of fluid in the barrel will be only at the level of the lowest amino acid there. So that becomes limiting. And if you raise the amino acid, for instance, lysine, then methionine becomes limiting. You see how it can vary depending on the ingredient composition. So we talk about the concept of limiting essential amino acids, and the ideal protein is the protein that we would ideally like to match the requirement of the fish. Now that's harder to achieve than it is on calculations and theoretical, because you have to formulate by computer, by using formulation systems, which do allow us to do that, and we aim to match the amino acid pattern of the fish, which is very close to the muscle tissue pattern of fish and the eggs and the ova. So this ideal protein was first put together for pigs in Aberdeen University in the UK. And if we express the amino acids as a percentage of the protein, then you see something very interesting. By normalizing this, we see that different species of fish, I'm sorry I don't have them all there, but you see fish like milkfish, the Japanese eel, the red drum, the common carp, the channel catfish, and the Chinook salmon. If you do it this way, you can see a lot of commonality, but they're not that different too much. So despite all their variations in quantitative requirements of protein, the amino acid makeup of that protein is fairly consistent. And that's not surprising, given that we are growing fish muscle, and fish muscle looks the same whatever species. Okay, I hope that is clear. And you can see some requirements for tilapia, and I don't want to be too bogged down with numbers, because the numbers can vary depending on the environment and the formulations, but these are general guidelines, okay? And this is the kind of data you will see in tables for different categories of weight class, fry to fingerling, fingerling to subadults, adults to broodstock. You see there that we see a trend which is downwards as the fish gets bigger. There's a relatively lower requirement for protein to energy ratio in the diets, and that's reflected in the data down there. Now, for first feeding fish, it's better to have a very high protein. We can afford to waste the diet a little bit, because FCR, feed conversions, is important, but not as important for the on-growing fish, where it's economically more important. but for first feeding fish, then we have diets with up to 50%, protein level, which is very high. And only certain types of ingredients can supply that, like, for instance, fish meal. So we are limited to the concentration of proteins that we can use. I hope that's clear, and I hope you're formulating some questions. And here we see with catfish, this catfish is the channel catfish, tilapia. These are American data. Catfish and tilapia use a wide variety of feed ingredients at higher temperatures. They can digest quite well at higher temperatures, these ingredients, and fried fingerling, 35 to 40% protein, sometimes even higher for the fry. Fingerlings to adults, 25 to 35, and broodstock fish do require more protein because they're producing over eggs, and so you must not treat brood fish like adult fish. They have to be specially looked after, and we have to cater for higher protein requirements for development of ova. Okay, and we see now that carbohydrates, as I mentioned earlier, are a very important component of the diet, especially for omnivorous fish. There's no fundamental requirement for carbohydrates because we can make carbohydrates from other sources, but carbohydrates in the feed would be a very good way of sparing the requirement to make them. So energy from carbohydrates is an important thing for fish to have in feed commercial diets, and we can provide that with different types of starches and grains. Grains and cereals are important sources, and by heat processing and everything we can make them more digestible. But digestibility and metabolism is similar to many mammals for omnivorous fish and different for carnivorous fish that require more protein and oils in their diets for energy sources. But ultimately, carbohydrates which are absorbed in excess are converted to lipids or stored as glycogen. So that's why you can get fat eating your Mars bars. You convert your sugar into lipid, into fat, and then you store that also as glycogen, which is mainly in the liver and muscle. And this is what carnivorous fish do. They store glycogen even when they don't need it because they don't know what to do with it. They actually produce excess sugar from gluconeogenesis. So there's a big difference between carnivores and omnivores. And you can see that carbohydrates in omnivorous warm water fish. I have an example, a little picture of digestion. Carbohydrates like starches have to be broken down to simpler sugar structures, like maltose, and ultimately to glucose. And the enzymes are present. They have these enzymes. Most of these warm water species can digest these. They have their amylases, they have their maltases, and they can assimilate the glucose quite efficiently. They also have the enzymes metabolically to phosphorylate the glucose to produce energy. Okay, so the omnivorous species. Carbohydrates in carnivorous fish, as I said, trout and salmon, just to be making some comparisons, they cannot assimilate carbohydrates, and that is also true for the warm water carnivorous species, like the cobias and the groupers and the other species, and especially eels at higher temperatures. We can grow eels at higher temperatures, faster growth. They are very carnivorous and don't do well at all on sugary diets. In fact, I don't either. I have to be very careful because I have a mild form of diabetes type 2. So I'm like a fish in some ways, especially a carnivorous fish. And here we see, interesting, I know it's a rainbow trout, but you can equate this to other species of warm water carnivores. And they have a less complex neural regulatory network so they don't have to worry about too much for sugar assimilation like we would have. But the poor use of glucose as an energy source is well known for trout and salmon. They do take in carbohydrates in their rich diet sometimes, but they can tolerate certain amounts, and they can be metabolically activated to utilize some of that sugar. But they don't have a lot of good control over their endogenous glucose production, and therefore they have a poor lipogenic ability to store excess glucose, and they lack an adaptive response to digestion absorption because they have a lower level of amylase, and more critically, they don't have this enzyme called a hexokinase or glucokinase to actually phosphorylate the sugar, and that's similar then in carnivorous marine species. Okay, Now then, fish nutrition, as you can see, involves so many components. It's really very, very much a multidisciplinary subject within its own discipline. Aquaculture, of course, is a multidisciplinary subject, but fish nutrition is also within itself a multidisciplinary subject. Now, I can't pretend to be an expert, although Hambai was very kind to state how much I am, well experienced in fish nutrition. Yes, I do have a very strong generic experience in fish nutrition, but professors must always learn. We never stop learning. And as he said to me, I'm still counting my students, counting the papers and subjects coming out. So I'm always learning. And the lipid nutrition of warm water fish is very interesting and less well known compared to the temperate cold water species like trout and salmon. We've studied, a lot of knowledge is out there on lipids and as a source of energy in structural roles, in building tissues, storage of fat, transport of fat-soluble vitamins. These are very important for that purpose. Lipids produce the most energy compared to other nutrient groups. So weight for weight, the lipids in the diets have a very high density of energy. Hence, that's why in salmon and trout diets, we have very high levels of oils and fats in the feed. That can be difficult sometimes when we make the diets under extruded conditions. But fortunately for us, it's a little bit simpler to understand in warm water species, but we still have a big knowledge gap, which we must address. And the nomenclature, the terminology of fats and oils and essential fatty acids is, of course, extremely complicated. And there are better people than me that know this area. And I have to give credit to my old University of Stirling in Scotland, where I did my PhD, because Douglas Tosher and his group there have done some extremely nice work over the years on the essential fatty acid pathways in mainly salmonid fish. And we can see here two groups of fatty acids, the omega-3s, which everybody will be familiar with, important in our diets for human nutrition, and the omega-6 fatty acid series. Now, without putting boxes around these too much, one could say really that the priority area in warm water fish nutrition, especially omnivorous fish and herbivorous fish, is the omega-6 fatty acid series, because it supplies the energy and supplies a lot of the requirements for phospholipid production and membrane structure, and the omega-6 contribute quite a bit to this. So we're talking about linoleic and arachidonic fatty acids in their metabolism, but we mustn't forget the omega-9 series, okay, the oleic fatty acid series, and linoleic, yes, and oleic are very, very important for that in the omega-9 series, C18-1, okay, so oleic acid, sorry. And so we have here two major pathways, and they are essential fatty acids for fish. And we know this to be true because some species of fish have lost the ability to elongate, to make the family of omega-6s and the omega-3s, to make the longer chain fatty acids. Hence, they become requirements in the diets. So the omega-6 family, you see, linoleic acid can flow into producing gamma-linolenic fatty acids, and then finally arachidonic fatty acids, the C20 with 4 double bonds, which are pro-inflammatory products. And we know that they can cause pro-inflammation in humans. And the omega-3 pathway is counter to that and is anti-inflammatory in its pathway mechanism. And metabolically, they are anti-inflammatory. So there's a balance, a critical requirements for both. And we must get that ratio correct in diet formulations. And the truth is, we don't really know too much about the significance in some species of fish. We do know that in higher level carnivorous marine species, there's a higher requirement for the omega-3 fatty acids, because here, these pathways here are not as efficient. So we need these pre-formed in the diet, especially in the larval stages during critical phases of development. Okay, so the jury is out here, and we need to do much more work. Now, warm water species like tilapia and carp have a higher levels of omega-6 fatty acids in their stored lipids, That's not to say we can't influence that by feeding omega-3 fatty acids in the diet. We can formulate diets to lipid tailor and put more omega-3s into the storage tissue of the fish. And that's some of the work that I'm doing at the moment, is to formulate diets to improve the image of tilapia, which does not have a very good image for omega-3s currently in normal production, because we use a lot of plant oils in the diets. Because fish oil, of course, is very expensive, and we are looking at algal alternatives to that. So the EFA, the essential fatty acid requirements, are important. Linolenic 18,3 N-3 is a precursor for N-3 HUFA, highly unsaturated fatty acid biosynthesis in many fish. Marine fish tend to have a requirement for N-3 fatty acids, especially the longer chain ones, C25 N-3, 22,6 N-3 in their lipid profiles. And salmon have a limited ability to extend that, so that is why we have this preformed requirement. Warm water fish have a lower requirement compared, but that's not to say that we can't, as I said, influence that. And the ratio of N-3 to N-6 and N-9 will play an important role in membrane modulation. Remember, warm water fish do not have to make their membranes more fluid because the temperature effect is quite dominant, whereas cold water species have to make their membranes more fluid. And the highly unsaturated fatty acids, of course, improve that. And here we see there, just to refresh, it's just a review, refreshment there. There are two lipid classes: polyunsaturated fats, soft, more mobile; saturated fats are harder. Some warm water fish will be able to digest more the saturated fats and the monoenoic fats. So poultry fats can be used in some fish, and lard and fats from the processing of from the rendering industry, which is something I'm very interested in. We use rendered proteins from animal byproducts like poultry protein and things like this, but we can also use some of the fats and oils from the rendering industry of some of the warm water species. Okay, and a deficiency of lipids can induce several pathologies. These are usually something you can measure and quantitate through histology. Ultimately, there's a problem of hemorrhaging in some cases, inflammation due to deficiencies, and you can induce this by taking them out of the diet and doing controlled experiments. And that's what people did in the past to define these requirements. And you would then have to formulate very precise semi-purified diets. It's very hard to formulate a diet to look specifically at individual fatty acids. They're very expensive to buy, and people have used radio labeling and stable isotopes to see what the requirements are and how the fish can metabolize. And you see there that a fish can actually store a lot of fat around the viscera. This is a problem, especially in some species. And during the winter phases, when temperature drops, some of the warm water species tend to divert their fat and energy stores into accumulating fat around the belly. And so this is an issue that can lead to other pathologies and stress. So we need to get that ratio correct, and carcass composition is important in processing and for the consumer. The consumer has complained in the past, and the retailer, about the excessive fat accumulation in certain species of fish, and including rainbow trout and salmon as well. And so this is a serious issue. And adding oils to the diets is not an easy thing to do in the feed mill, because of being in a liquid state, being many of them are oils. It's hard to meter those properly. We are lucky that many of the omnivorous warm water species, the diets are cold pelleted or using pelleting systems, not technically as extruded. There are some extruded diets, of course, for tilapia, but adding high oil levels to extruded diets for salmonids is a challenge. But it's been overcome by the industry. But it's not the same. It's not as simple as extruded diets that we use for most of the other species. Tilapia, people do use expanded diets for tilapia, semi-floating diets and things like this, semi-sinking. But in the main, there are typical pelleted feeds. And then lipids for tilapia, again, as I said, they are lower in the 8 to 12% in the diet, typically. Many of the raw materials can supply that, but we need to top it up with plant oils. And I like to put a little bit of fish oil in there, just a little bit, to make sure we do have some omega-3s. But of course, the tendency is not to do that. And diets for tilapia can be fish meal-free, fish oil-free. So we have to expect the composition of the fish to reflect that. They can be met by omega-6, omega-9 fatty acids for their metabolism. And although low in omega-3, as I said, we can enrich the fish with oil from high in EPA. Sorry, that should be oil, not oiled. High in EPA and DHA fatty acids. Okay, and that's a project I'm working on right now. And vitamins in fish nutrition, well, like in every animal system, they require a complement of vitamins which can be designated fat soluble or water soluble. I don't like those terms really, but water soluble. Most of the water soluble vitamins are actually bound as complex organic structures. They're not as water soluble as you might think. But the problem is some of those vitamins are thermolabile and can be influenced by processing of the diet during the milling stages, the grinding, and some of those vitamins you would not actually rely on coming from natural raw materials. So all these vitamins and other trace elements of vitamins and minerals will be found in mineral and vitamin premixes. And that's a big art in itself, to formulate diets for different species of fish and to target fish is very much scientific, and a lot of research goes into designing the vitamin premixes to make them more stable and to compensate for any losses that are associated with thermal processing and pelleting. Of course, extrusion technology is much more damaging to vitamins because of the higher temperatures and pressures in the extruder. So we would rely on vitamin premixes, and but some fish farmers would add certain vitamins to top dress the pellet. For example, vitamin C is often added to the diets at the farm level by certain farmers because vitamin C is a very good vitamin and is a good prophylactic treatment as well. So four fat-soluble vitamins, vitamins A, D, E, and K, are very important in the feed for fish health. And warm water fish will have a higher metabolic demand, so those vitamin and mineral requirements will be proportionate to the higher energy. So we would ideally formulate the diets to be compensating for the higher energy intake. And we may find more vitamins and minerals in the diets to compensate for losses. Okay, so trace elements, I've done a lot of work. I did work on sea bream. One of my former students, Paul Morris, who works now for Maui Aquaculture, formerly Marine Harvest. He did a lot of work to define the vitamin requirements for the sea bream. And sea bass and sea bream are those fish in the middle ground. They do better at higher temperatures, because if you have a sea bass growing in the waters around the UK, they will take a long time to grow. But the Asian sea bass, the barramundis and all those, they are obviously will function better in around the 20 degrees Celsius. And so it's very important to get that right for these fish. Okay, so leaching is an issue, of course. We might lose water-soluble components of the diet in the feed, so we have to use binders, and we have to consider the stability of the diet. And this is very important in shrimp farming, of course, as well, when the diet is in residence for a long time in the water. Fortunately for us, most of our fish have good appetites and eat the diet very quickly. So we don't have to worry too much about that. Now here we are. Here are the fat-soluble vitamins in detail. We use international units per kilogram of diet. This is the requirement, and we have to then formulate vitamin premixes to make sure that requirement is met fundamentally through our knowledge base. And our knowledge base comes from scientific papers, from data like you find in the NRC 2011 requirements for fish and shrimp, which I'm afraid you have to be careful of because some of that data is very old now. 2011, there's been a lot of research since then. And so I know that vitamin E research is undergoing in many countries now at the moment, because vitamin E is a component of cell membrane stability and structure. Vitamin E, very important antioxidant. and will become even more important as some of those chemical antioxidants, the legislation will prohibit them from being used. Okay, so in the future, we'll have to look more closely at natural vitamin sources like vitamin E. Vitamin A and vitamin D are vitamins that have a role to play in genomic expression. These vitamins can actually influence the development of fish and are critical in the early stages of development. And so we must look again at vitamin A in terms of antioxidant role and function. And vitamin D is, apart from being important in bone health and skeletal structure health, has many other roles to play, and some of them have hormonal roles, like vitamin D and vitamin A. The water-soluble vitamins are a suite of vitamins. I like to look at them together, not as individuals, because they play a synergistic role. But riboflavin, pantothenic acid, niacin, vitamin B12, biotin, folic acid, thiamine, and vitamin C are all part of these water-soluble components, and you will find them in different forms, and companies, big companies, international companies, specialize in vitamin production, and you will not find them just as the chemical. They'll be bound to other things and stabilized. Okay, and that will affect their bioavailability as well. It's important to look at the bioavailability of each of those in the premix. And choline is usually added separately, and myo-inositol, these are usually added separately to the diets because of their properties, functional and physical chemical properties. And there you see some of the requirements for minerals, the macro elements, calcium, potassium, chlorine, potassium, electrolytes, of course. We don't tend to think of them as they're not trace elements, they're macro elements. But when we get down to sort of phosphorus and calcium, those are very critical for bone and skeletal structure and development there. And phosphorus, of course, is an important metabolic role in phosphorylating compounds, as we mentioned, phosphorylating sugars and things like that. So we must bear the calcium phosphorus requirements have an optimum ratio themselves. Too much calcium can affect phosphorus requirements. Too much phosphorus can interfere with calcium absorption. Fortunately, in the marine environment, fish get a lot of calcium. But in the freshwater environment, where most of our warm water species are important, like tilapia, carp and everything, we must be careful about phosphorus. And so don't underestimate that. And as we replace fish meal with other sources which are lower in phosphorus, the phosphorus requirement becomes even more important to study. Okay, I'm looking at my time here, and I think we're doing all right. Yes, coming up to the end, almost. It's a full hour. But I've been engaged a lot with trace element nutrition and zinc and selenium, particularly. I've done quite a lot with that. And for warm water species like tilapia, we know less than we do for salmonids. We do need to open our windows there a bit more to understand the role of selenium and zinc. Zinc is an important one because zinc oxide has been used a lot in the pig farming industry for controlling bacterial population in the gut, and that's going to be taken out soon. So people are looking at zinc sources for other animal species as well. And then we see tables like this. These are guidelines. I don't want you to be fixed on numbers, but it gives you a guideline to the types of requirement levels that are required for tilapia here. In particular, an example for tilapia, it reiterates what I've been saying in the other slides. It just confirms the major macro requirements for nutrients and scanning protein, lipids, carbohydrates, vitamins, and minerals. But always remember that your fish must relate to the environment they're in, and these requirements can vary, and different feed companies will have preferences, they'll have different specifications, but they broadly match that table for tilapia. And there you see the same kind of thing again. You can see that the requirement for lipids is quite low compared to the requirements we would have for other species of fish, okay, especially trout and salmon. And you see there that they highlight the lysine methionine requirement. They stand out because they are rate limiting. Okay. Right. Here is, I'll be coming to the end soon, but here are some formulations. These are just sources of formulations. There are many companies formulating diets for tilapia around the world globally. I'm aware that the audience is very global and that we have different raw materials in different parts of the world. But you can see here that in this particular example here, fish meal is quite prominent. It's 15% for the pre-starter diet. It drops down to around 5%, and very often now we see fish meal free diets. We see fish oil being used moderately, but we have to look at maybe replacement for that in the future. But the warm water species are not as greedy for fish oil as, say, the salmon and trout industry. Corn, rice bran, wheat bran, these are cereals which provide the carbohydrates, but we put more and more of those into the on-growing diets where the fish can tolerate that. Soybean meal, well, that's an interesting one because the sustainability of soybean meal is also questionable compared to other ingredients. And people are asking about soybean meal, whether we should be using more and more in fish feeds. So you can see there's a trend to try to limit that to some degree. But fortunately for us, there's a lot of possibilities out there for us to formulate the diets. There's no rules and regulations about what we use. It's open to flexibility depending on price, depending on where you are in the world. So when people ask me what's a good tilapia diet, I can tell them there's 101 plus good tilapia diets. But you have to look at what matches best your area of the world. Okay, and cost, of course, these are in US dollars. I use a system called linear least cost formulation. I have that software on my computer. I can formulate diets and play around with the prices and try to match those requirements for the species in question. It's fun. I enjoy doing that. Sometimes I'd like to throw something at my computer because it doesn't always behave. It gives me crazy results. But then it's probably a crazy professor putting the data in the first place. And then you see another, this is one that was produced by a company in China. It's an example of a formulations for tilapia, Nile tilapia in China. You see there high levels of fish meal, almost half the diet was fish meal for their fry. And then it drops off quite a lot for the grow out. And it's very similar. And if you see there, they make sure that they get their vitamin and mineral premixes are usually around 0.5 to 1%, sometimes higher for the formulation. And that will depend on the concentration of the components of the vitamin and mineral premix. And you can see there that as they reduce the fish meal to a high extent on the far right, you see they have to add methionine to balance the requirement for essential amino acids. Okay. And so you see there at the bottom there, the approximate composition, the formulation has the protein dropping down to about 28% for a grow out feed. And that's quite typical. And the prices, of course, are there in US dollars per ton. But of course, there'll be a markup there for profit as well. Don't quote those prices. That's just a guideline. So I hope in this hour you have been able to appreciate the complexity involved, the challenge of working on fish nutrition for different species of fish, let alone the different domains, warm water versus cold water versus freshwater, brackish, and full-strength marine. All those things, the environment will play a dominant role in many ways. So I hope that you'll see it as not just a standalone discipline. It's very open to research and development, and I'm very pleased to be part of that. And it keeps me young and fit, I hope, and busy. And as Hambai said very nicely, I'm still counting the work and the students. I'm not doing as much as I used to, but I think I'm doing more now in other areas and working more and closer with industry. And that is very interesting and nice to be able to engage. So I hope that hour was productive and it was recorded. So if you missed something, I'm sure you will be able to play me back. And if you were hoping to have President Biden, you made a mistake. You chose the wrong channel. It's not the Democratic Convention. Thank you very much.
Okay, thank you very much, Professor Davis. So everybody, thank you very much for attending today's wonderful webinar by Professor Davis. Professor Davis, again, we thank you very, very much for your expertise and your excellent presentation. And it was very nice to be back in school again.
Yeah, well, I have to give you now your exam results. I have to grade you all now.
Okay, so if you have any questions, please contact your local representative, Pathway Intermediates representative, and we will get back to you with your questions or comments. Now, everybody, thank you very much for attending today's webinar. I will hope to see you in the near future, and look out for that announcement by Pathway Intermediates. Thank you, everybody. Have a wonderful day.
Thank you very much, and good health to all of you.